Hello, everybody, and welcome to the PhD course on basal metabolism and molecular mechanisms in diabetes, held by the Danish Diabetes Academy. My name is Niels Fergerman, and I'm a professor in biochemistry at the University of Southern Denmark. Today, I will give you a short introduction to how fatty acid metabolism is regulated, and obviously also how carbohydrate metabolism is regulated. So to begin with, I will try to give you a very brief overview of what we need to understand and what we need to focus on in order to understand how lipid metabolism and carbohydrate metabolism is regulated. So I will initially give you an overview of how these two parts of metabolism are connected to each other, and then later focus on how glycolysis and gluconeogenesis are regulated, and of course also touch upon how glycogen metabolism is regulated. Later on, and during when talking about carbohydrate metabolism, we of course also have to understand how it interacts with lipid metabolism. So I will touch upon how lipolysis is regulated in adipose tissue, and later on also touch upon how fatty acid degradation by beta oxidation in the mitochondria, and also how fatty acid synthesis is regulated. So to begin with, we need to understand how we deal with two of the primary energy substrates that we use on a daily basis. And this is, of course, glucose and fatty acids. And fatty acids, as you know, they will be bound primarily to albumin. So we need to understand how we actually regulate the levels of glucose and fatty acids in order to control how the energy expenditure in the different tissues and systemically is regulated. And of course, we know that we should try to keep the glucose concentration constant around 5 millimolar. So basically, we try to do that by regulating how much fatty acid we actually use as an alternative energy source instead of using glucose. So we need to understand how we actually handle glucose and fatty acids in order to keep the glucose concentration constant around 5 millimolar. And this type of interaction between fatty acid and glucose metabolism is also known as the glucose fatty acid cycle or the Randle cycle. We know that under certain conditions, muscle can actually use quite a lot of glucose. Up to 80% of the total use of glucose can actually occur in muscle. So obviously, if we can control how much glucose that is used in the muscle, we can also save quite a lot of glucose for other tissues. So we need to understand how glucose metabolism is regulated in muscle, and how fatty acids and maybe also ketone bodies can interact with glycolysis, for instance. We, of course, also need to know that the liver plays a significant role in keeping the glucose concentration constant by, and the liver can, as we know, can produce glucose available for the circulation by either degrading glycogen, or after degradation of glycogen, the liver can also produce glucose via the gluconeogenesis. So the liver obviously plays a very important role in maintaining constant glucose levels in the circulation. So we need to understand what occurs in the liver in order to know these regulatory mechanisms. Obviously, we also need to look at how fatty acid metabolism is controlled. And we all know that fatty acids are stored as triglycerides in the adipose tissue. And the degradation of triglycerides by lipolysis is heavily regulated. and we need to understand how how lipolysis is regulated in order to understand how we control the concentration of fatty acids in the circulation. So obviously we need to know what goes on in the adipose tissue as well. Importantly is sort of the master regulator of metabolism, which of course takes place in the brain by our nervous systems. And there are some highly specialized mechanisms that are capable of sensing how much glucose that are available in the circulation. So once the brain can sense that the glucose concentration is changing in the circulation, it will signal to other tissues that then will try to cope with these changes. So the brain is, of course, also extremely important, also because the brain by itself can use a lot of glucose. And finally, we of course also need to include the pancreas, which, as you know, produces many different important hormones, especially in this case, insulin and glucagon, that obviously plays important roles in the regulation of glucose levels in our circulation. So we need to understand what actually goes on in these different tissues in order to be able to control the glucose level as well as the fatty acid level in our circulation. So if we, for instance, take an example where we go from a fed state to a fasting state, then we can try to define or look at what actually happens in our circulation with respect to the glucose level, but also with respect to the fatty acid level, and actually also what happens in the tissue when we go from a fed state to a fasting state. So when we go from a fed state to a fasting state, we know that the glucose level will decrease. Not very much necessarily, but enough so the brain actually can sense that the glucose level is changing. In this case, it may decrease a little bit. So the brain will sense these changes, and via the sympathetic nervous system signal to the adipose tissue that it should activate production of an alternative energy source. And obviously this will be degradation of triglycerides to fatty acids via lipolysis. So when lipolysis becomes activated, we know that fatty acids will be secreted from the adipose tissue, which will or will result in an increase in fatty acids, in the concentration of fatty acids in the circulation. This will, of course, lead to an increased flux of fatty acids to the muscle, but also to the liver, which, of course, also will increase the degradation of fatty acids in the muscle to produce more ATP. Interestingly, there are metabolites coming from the degradation of fatty acids that actually inhibit the oxidation of glucose. So simply by increasing the fatty acid concentration in the circulation will actually result in inhibition of glucose oxidation in the muscle. At the same time, the liver will start producing glucose in order to keep the glucose level constant. So not only we spare glucose from being oxidized in the muscle, but at the same time, the liver will produce glucose to the circulation, so we are able to maintain a constant glucose level. And in this case, the liver will start degrading glycogen, so we actually produce glucose units for the circulation. Later on, when we have used the glycogen stores in the liver can produce glucose via gluconeogenesis. This will, of course, result in a slight increase in glucose, which is enough to signal to the pancreas that insulin should be secreted from the pancreas, and insulin is a strong inhibitor of lipolysis. So this will obviously result in inhibition of lipolysis. At the same time, the brain can sense that the glucose level is slightly increasing, and this will obviously result in a decreased activation of lipolysis from the sympathetic nerve system, overall leading to a decrease in lipolysis, and this will obviously result in lower concentration of fatty acids in our circulation. This will again result in a low flux of fatty acids to the muscle and to the liver, and will relieve the inhibition of glycolysis from these metabolites from fatty acid oxidation, resulting in increased oxidation of glucose. And this sort of cycle will happen all over again, because if we imagine that glucose now can be oxidized again, that will result in decreasing glucose concentrations, and then we will activate some of the same mechanisms that the brain will sense that the glucose level is decreasing again, will increase activation of lipolysis, and the fatty acid concentration will increase, and then the flux will increase of fatty acids to the muscle and to the liver, and then it will kind of happen all over again. And this is exactly what is meant by the glucose fatty acid cycle. We also know that the fatty acids that have not been used, fatty acids that are in excess, will be transported to the liver and be incorporated in triglycerides again, which will be packed into lipoproteins like VLDL particles, which will be transported to the circulation and transport the fatty acids back to the adipose tissue. So that when the glucose concentration is increasing and the fatty acid concentration is decreasing, the excess fatty acids will, as I said, be incorporated into lipoproteins and return to the adipose tissue. And the increasing glucose concentration will, of course, also support the re-esterification of fatty acids into triglycerides again. So in order to understand how these different mechanisms actually take place at a more molecular level, we need to understand what actually goes on in the adipose tissue, in the muscle, and in the liver. And if we also extend these regulatory mechanisms to go past 24 hours, where the glycogen stores have been used in the liver, we will see that the concentration of ketone bodies will increase, and the ketone bodies will can be used by different tissues like the brain and also the muscle as an alternative energy substrate. But the ketone bodies can actually also signal to the pancreas that it should increase insulin production and secretion, and thereby trying to control how much fatty acids are being released from the triglyceride stores in the adipose tissue. Interestingly, the ketone bodies can also, as fatty acids, can produce specific metabolites when they are being oxidized in the muscle that will inhibit glucose oxidation. So even after 24 hours where we start producing ketone bodies, these ketone bodies can also control not only release of fatty acids from the adipose tissue, but actually also how much glucose that is being oxidized in the muscle, thereby trying to control the glucose level again under these extreme conditions. And also in the brain, the ketone bodies can inhibit glucose oxidation, thereby sparing the glucose for other important tissues. So in order to understand how these molecular mechanisms take place in the different tissues, we have to understand what goes on a molecular level in the different tissues. And the following parts of the lecture, I will try to dig into some of these regulatory mechanisms in adipose tissue, in the muscle, and also in the liver, and try to look at how these different mechanisms in specific tissues actually can affect each other across these different tissues. So if we start by looking at how lipolysis is regulated in adipose tissue, then we can take one of these classical examples from a textbook, in this case, Linear Principles of Biochemistry. Then we can see that a receptor can pick up a signal from a hormone. And in many textbooks, it's shown that glucagon from the pancreas can actually activate lipolysis in adipose tissue. I like to stress that this is only taking place under extreme conditions where the glucagon level is very high. Normally, or in vitro, glucagon can indeed activate lipolysis, but in vivo or under in vivo conditions, glucagon will not activate lipolysis, but instead it will be norepinephrine or noradrenaline coming from these sympathetic nerves that directly innervate the adipose tissue, that these nerves will produce noradrenaline, which will activate a G protein coupled receptor, and via a stimulatory G protein activate this enzyme called adenylate cyclase that produces cyclic AMP, which you know will activate protein kinase A that then will phosphorylate different enzymes involved in lipolysis. One of the enzymes that is being phosphorylated by protein kinase A is the enzyme called hormone sensitive lipase. So once this enzyme has been phosphorylated by pKa, it will actually try to get access to the lipid droplet where all the triglycerides are stored. But it can only access the lipids after another protein has been phosphorylated by protein kinase A, and this protein is known as perilipin. So when perilipin has been phosphorylated by pKa, it will allow the hormone-sensitive lipase to access the lipid droplet. But the hormone-sensitive lipase has primarily activity against diacylglycerols. So that means that we need another lipase in order to inhibit triglycerides. This lipase is known as ATGL, or adipocyte triglyceride lipase, which is activated by a small protein called CGI. CGI normally sits or interacts with perilipin, where it's kept or where it's controlled. But once the perilipin is phosphorylated by protein kinase A, perilipin will relieve CGI that will allow CGI to interact with ATGL and thereby activate ATGL. So now we can not only degrade diacylglycerols, but also triacylglycerols. So when we activate protein kinase A by increasing cyclic AMP levels, we will not only see that perilipin is being phosphorylated and thereby allowing CGI to activate ATGL, but we will also see that the hormone-sensitive lipase becomes phosphorylated and becomes activated and will start degrading diacylglycerols. So once we produce or once we degrade triacylglycerols and diacylglycerols, we will of course produce fatty acids, and we'll get another fatty acid by degrading monoacylglycerol to fatty acids and free glycerol. And free glycerol and the fatty acids will be secreted by the adipocyte to the circulation. And this is simply the reason why we see an increase in the concentration of fatty acids in our circulation when we enter a fasting or starvation situation, simply because the level of noradrenaline increases in our in or around the adipocyte. So it's important to stress that it's not glucagon, but it's noradrenaline that actually activates the receptor and leads to activation of protein kinase A that then will activate lipolysis. And this will increase the concentration of fatty acids in the circulation. And these fatty acids can then be transported to other tissues that can use fatty acids as an energy substrate, for instance, skeletal muscle cells. All right, so now we know how the fatty acids are released from the adipocytes. And these will, of course, enter the circulation, as I said, and eventually reach some of the target tissues, for instance, myocytes. Then we know that fatty acids from albumin can be imported via specific transport proteins into the myocytes, where the fatty acids need to be activated to what we know as acyl-CoA esters in order to enter any kind of metabolism. This activation requires ATP in order to activate the fatty acids. And once we have activated the fatty acids, we know that these fatty acids then are being transported to the mitochondria via the CPT1 system, where they will enter and be degraded by the beta oxidation, which will produce lots of acetyl-CoA and also NADH and FADH2. We also know that glucose can enter the myocytes via specific transporters, in this case, GLUT4. And once glucose has been imported into the myocyte, the glucose will be phosphorylated by the first enzyme in glycolysis, known as hexokinase. Glucose will then be phosphorylated to glucose 6-phosphate, which then will be converted to fructose 6-phosphate, which then, by the activity of phosphofructokinase 1, can be further phosphorylated to fructose 1,6-bisphosphate. This can then be converted to PEP, which then can be converted to pyruvate by pyruvate kinase. Pyruvate can now enter the mitochondria, and in the mitochondria, pyruvate can then be converted via the activity of pyruvate dehydrogenase to acetyl-CoA. And we know that, and this is actually an extremely important point, that the acetyl-CoA units and NADH can inhibit the activity of PDH, the pyruvate dehydrogenase. We also know that these same two substrates can promote the phosphorylation of pyruvate dehydrogenase, which once the enzyme has been phosphorylated and allosterically inhibited by these two substrates or these two metabolites, the enzyme becomes almost completely inactive. And this is extremely important because that will secure that the glucose is not converted to acetyl-CoA, and it will also, of course, promote that the acetyl-CoA from the fatty acids can be oxidized in the TCA cycle. Because the acetyl-CoA, once the acetyl-CoA concentration increases just a little bit in the mitochondria, it will activate an enzyme called pyruvate carboxylase, thereby producing oxaloacetate. And we know that along with acetyl-CoA, oxaloacetate can produce citrate. So once the fatty acids enter the myocyte and are oxidized in the mitochondria, the acetyl-CoA level will increase, and so will the oxaloacetate level. This will result in an increase in the citrate concentration, which can be transported out of the mitochondria and then actually result in inhibition of the phosphofructokinase 1, which of course will result in an increase in fructose 6-phosphate. And because this is an equilibrium between fructose 6-phosphate and glucose 6-phosphate, the level of glucose 6-phosphate will also increase, and that will result in a strong inhibition of hexokinase, thereby inhibiting oxidation of glucose. So simply because the fatty acid concentration in the circulation increases during starvation, that will result in an increased influx of fatty acids in the myocyte and an increased oxidation of fatty acids in the mitochondria, and then simply result in an increase in citrate levels that eventually will increase fructose 6-phosphate level, because the phosphofructokinase 1 enzyme is inhibited by citrate, and that will result in an increase in glucose 6-phosphate levels, and then inhibition of hexokinase. So simply by increasing fatty acid concentration will actually lead to an inhibition of glucose oxidation. Obviously, glycolysis is also regulated by fructose 2,6-bisphosphate levels in the muscle, as we know from the liver, which I will come back to later. And we also know that glycolysis can be regulated at the level of pyruvate kinase by ATP, acetyl-CoA fatty acids, and amino acids, which all inhibit pyruvate kinase. And we know that fructose 1,6-bisphosphate can actually also activate the enzyme. But all these different mechanisms will basically result in a strong interaction between fatty acid metabolism and glycolysis. And in a case where we are looking at glycolysis or the regulation of glycolysis in muscle during fasting, we know that the fatty acid concentration increases in the circulation, which overall will result in increased citrate levels that will inhibit phosphofructokinase 1 and later on hexokinase as well, leading to an inhibition of glucose oxidation. Of course, we cannot degrade fatty acids without having a little bit of pyruvate, because we need production of oxaloacetate in order to produce citrate and to keep TCA going, or the citric acid cycle going. So what we actually do here is we are trying to use less glucose coming from the circulation, but we need some oxidation of some glucose in order to produce pyruvate. And this glucose comes from oxidation or degradation of glycogen to glucose units. So if we try to keep it simple and try to look at, from a helicopter point of view, look at what actually goes on with glycogen. We know that UDP glucose, an activated form of glucose, is the substrate for glycogen synthesis. And glycogen can then be degraded typically to glucose 1-phosphate, which relatively easy can be converted to glucose 6-phosphate and thereby enter glycolysis. We know that the enzyme that we need in order to produce glycogen is called glycogen synthase, which also can be phosphorylated and become less active. We know that glycogen can be degraded by an enzyme called phosphorylase, which also can be phosphorylated. And in this case, when phosphorylase becomes phosphorylated, it will be activated. Phosphorylase will be phosphorylated by an enzyme called phosphorylase kinase, which also is activated by phosphorylation. So these are more or less the scaffold we need in order to understand how glycogen metabolism is regulated. We know that under conditions like fasting or perhaps exercise, that the level of adrenaline will increase in the circulation. As we saw in the adipocytes, adrenaline, like noradrenaline in adipocytes, can activate a G protein coupled receptor, which also via a stimulatory G protein can activate adenylate cyclase that produces cyclic AMP from ATP. Once the level of cyclic AMP increases, it will, as we saw in adipocytes, also activate protein kinase A. And we know that protein kinase A can activate or can phosphorylate glycogen synthase, leading to a less active form of glycogen synthase. But especially during exercise, we also know that we have other regulatory mechanisms that can control glycogen metabolism. And this comes from the production of acetylcholine, a neuromessenger coming from motor neurons. So once we stimulate contraction of muscle, we can do that by secreting acetylcholine from motor neurons, which will, via an acetylcholine receptor, which also is a sodium channel, actually produce an actual potential leading to depolarization of the plasma membrane. This will then affect a number of proteins, for instance, the dihydropyridine receptor, which interact with another protein in the sarcoplasmic reticulum known as the ryanodine receptor. The ryanodine receptor is also a calcium channel. So once we have the action potential and depolarization of the plasma membrane, this will actually lead to a release of calcium from the sarcoplasmic reticulum to the cytosol, leading to an increase of calcium in the cytosol. So along with the activation of protein kinase A and the increase in calcium concentration, we will see that phosphorylase becomes activated. Protein kinase A can phosphorylate two subunits, the alpha and beta subunits of phosphorylase kinase, and the calcium ions can bind to a subunit of phosphorylase kinase, which is a delta subunit, which also is calmodulin. So once we activate protein kinase A by adrenaline and increase calcium concentrations in the cytosol by stimulating production of acetylcholine, we will see an increase in the activity of phosphorylase kinase. The activation of phosphorylase kinase will then lead to an phosphorylation of phosphorylase, leading to a more active form of phosphorylase, which then will start degrading glycogen to glucose 1-phosphate. And at the same time, we have inhibited glycogen synthase by phosphorylation by, for instance, protein kinase A, which, as I said before, leads to a more inactive form of glycogen synthase. So we not only activate degradation of glycogen, but we actually also inhibit the synthesis of glycogen by these mechanisms. We also know that there are other activators of glycogen or regulators of glycogen metabolisms. For instance, when we release calcium from the sarcoplasmic reticulum, it will, as you know, increase or stimulate muscle contraction, and the muscle contraction by itself will also use quite a lot of ATP. In order to, or when ATP is produced or is used, ATP is converted to ADP. And in order to produce some of ATP again, we can actually use 2 moles of ADP to produce 1 mole of ATP, and that will obviously also result in production of AMP. So we basically use 1 ADP to phosphorylate another ADP, leading to production of ATP and AMP. That means that the level of AMP will increase actually quite dramatically during muscle contraction. And AMP is a strong allosteric regulator and stimulator of the non-phosphorylated form of phosphorylase. So simply by increasing the level of AMP, we can stimulate degradation of glycogen further by stimulating the activity of the non-phosphorylated form of phosphorylase. Then we also know that it's quite important to be able to regulate the phosphorylation of the different enzymes, that being glycogen synthase, phosphorylase, and also the phosphorylase kinase. We know that the enzyme that can dephosphorylate these enzymes is the enzyme called PP1 or protein phosphatase 1. This is being regulated by protein kinase A. So under these conditions, for instance, during fasting or exercise, we know that protein kinase A is active because we have activated the production of cyclic AMP, and the activation of protein kinase A will actually lead to an inhibition of protein phosphatase 1 that obviously, when inhibited, will not dephosphorylate glycogen synthase. It will not dephosphorylate phosphorylase and not dephosphorylate phosphorylase kinase, leading to or that will, of course, keep the enzymes active or inactive in terms of the glycogen synthase. So these control mechanisms will, of course, ensure that under these conditions that glycogen is being degraded and not being synthesized. Then we also know that insulin will, of course, also have a major impact on glycogen synthesis and degradation. We know that insulin, via its insulin receptor and some of the downstream effectors, can lead to an inhibition of the enzyme called glycogen synthase kinase 3. And this is another kinase that can phosphorylate glycogen synthase and as PK also inhibit the enzyme. So in this case, where we are fasting or exercising, the insulin level will be low, leading to or that will overall result in an active glycogen synthase kinase 3, and thereby we actually will keep the glycogen synthase phosphorylated. And then there's another enzyme called the calmodulin-dependent protein kinase, which is activated by calcium. And in this case, we actually have already increased the concentration of calcium in the cytosol, which not only will affect the activity of phosphorylase kinase, but also bind and activate the calmodulin-dependent protein kinase. And thereby we see further phosphorylation of glycogen synthase, which again will lead to a further inhibition of the enzyme. So again, all these different mechanisms will actually control that we're not synthesizing glycogen, but actually degrading glycogen. And if we look further into how the PP1 or protein phosphatase is regulated, then again, if we look at this part of the figure, we know that protein kinase A can actually inhibit protein phosphatase 1. If we look into the details of this regulation, we know that either adrenaline, also known as epinephrine, or glucagon will lead to activation of protein kinase A. pKa, or protein kinase A, can phosphorylate the regulatory subunit known of PP1, known as the GM subunits of PP1. So once GM becomes phosphorylated, the active or the catalytic part of PP1 will dissociate from the regulatory part and become less active. But it's still a little bit active. So in order to inhibit it further, protein kinase A will phosphorylate an inhibitor protein, which will, once it's phosphorylated, bind and inhibit PP1, or the catalytic subunit of PP1 further. So we completely inactivate protein phosphatase 1, which, as I said before, will lead to that glycogen synthase, or the phosphorylated glycogen synthase, or the phosphorylated form of glycogen synthase, is not being dephosphorylated and thereby activated. So we keep it as the phosphorylated form, as we do with the phosphorylase and phosphorylase kinase. So this step is actually quite important in order to control how the enzymes involved are being kept as phosphorylated. So we, of course, also have to understand what happens in the liver. So we have now looked at how glucose metabolism is regulated in muscle cells, but in liver, we see some of the same mechanisms as we saw in muscle, but there are also other mechanisms that are a little bit different. So when we look at glycogen metabolism in the liver, we have, as we saw before, some of the same pathways. UDP glucose is substrate for glycogen synthesis, and glycogen can be degraded to glucose 1-phosphate, which again can be relatively easy converted to glucose 6-phosphate and thereby enter metabolism. We also know that glycogen synthase is phosphorylated and becomes less active. And so with phosphorylase, as before, phosphorylase can be phosphorylated and become active. and it requires the activity of phosphorylase kinase, which also is activated by, for instance, phosphorylation. So the overall scaffold is more or less the same as we saw before. In the liver, we know that glucose is primarily entering the hepatocytes via a glucose transporter known as GLUT2. This transporter has a high Km, meaning that the glucose concentration in the circulation will more or less be the same as the concentration in the inside of the hepatocyte. So the glucose is more or less in equilibrium across the membrane. Thereby, that when the glucose concentration increases in the circulation, the glucose concentration in the hepatocyte will also increase. In the liver, we know that glucagon, as adrenaline in muscle cells, can activate a G protein called receptor, known as the glucagon receptor, which interact and can activate a stimulatory G protein, which again can activate the adenylate cyclase, leading to increased levels of cyclic AMP and activation of protein kinase A, which, as we saw before, can phosphorylate glycogen synthase, leading to a less active form of the enzyme. In liver, adrenaline can also regulate calcium levels. As we saw in muscle, calcium levels could increase upon activation of, for instance, contraction. But in the liver, we know that adrenaline via a G protein called receptor, in this case an alpha adrenergic receptor that interacts with a GQ protein, that again activate a phospholipase called PLC that then degrades phosphoinositol bisphosphate to diacylglycerols and IP3. IP3 can bind to an IP3 receptor that is in the same family as the ryanodine receptor as we found in muscle. But once IP3 binds to the receptor can actually open as a calcium channel, and then we will see that calcium efflux from the ER to the cytosol. So simply by simply by increasing the level of adrenaline in the circulation will also lead to an increase in the calcium levels in the cytosol in hepatocytes. So as before, we will see that protein kinase A will phosphorylate phosphorylase kinase at the alpha and beta subunit and thereby activate the enzyme, but the enzyme becomes only 100% active once it has bound calcium ions to the delta subunit. Once we have activated phosphorylase kinase, we will see that this enzyme will phosphorylate phosphorylase, which then will become active and degrade glycogen to glucose 1-phosphate units. Here we know that it's not pKa as we saw in muscle, but it's actually glucose that will stimulate the activity of PP1, which again can dephosphorylate the enzyme. And this actually makes good sense because if the concentration of glucose increases in the circulation, it will also increase in the cytosol of the hepatocyte. That will lead to an activation of PP1 that will dephosphorylate the enzymes involved in glycogen synthesis and degradation. And if PP1 becomes activated, glycogen synthase will become more active and phosphorylase will become less active. And that will turn the situation around. So we'll not degrade glycogen, but we'll actually start producing glycogen. So simply because glucose levels eventually will increase, we can actually control how much glycogen that is being synthesized and of course also being degraded simply by regulating the activity of PP1, which I'll come back to later. We also know, as we saw before, that insulin can inhibit the activity of glycogen synthase kinase 3. And if we are in a fasting situation, insulin concentrations will be low, leading to an active form of glycogen synthase kinase 3, which will phosphorylate glycogen synthase and make the enzyme less active. So in a fasting situation where the insulin levels are low, glycogen synthase kinase 3 or GSK3 will be active, leading to a further inhibition of glycogen synthase. Then we also know that if we see an increase in the circulation, in the glucose concentration in the circulation, that will also lead to a production of glucose 6-phosphate that can activate even the phosphorylated form of glycogen synthase, leading to an increased activity of the enzyme and stimulate glycogen synthesis. Interestingly, we also know that glucose 6-phosphate can actually inhibit phosphorylase, which obviously also makes good sense, because if we increase the level of glucose 6-phosphate, we are not interested in degrading glycogen further. So it makes actually good sense that glucose 6-phosphate coming from glucose will inhibit the enzyme. And the activity of phosphorylase can actually also be regulated by the energy charge, meaning the ratio between ATP and AMP. All right, so if we for a moment return to the regulation of PP1 by glucose, this mechanism is actually quite important in order to understand how glycogen metabolism is regulated in the liver. As you probably can recall, that regulation of PP1 is different in muscle. So in the liver, as I said, PP1 is controlled by glucose and not by protein kinase A. So in the liver, we will see that the PP1, the catalytic subunit, depicted here by PP1, and its regulatory subunit known as GL, will actually interact with glycogen phosphorylase. So once these, when these proteins interact, the PP1 will be less active, or actually be inhibited by phosphorylase. So the phosphorylated form of phosphorylase will actually inhibit PP1 activity. Interestingly, when the glucose concentration increases in the circulation, the glucose concentration in the hepatocyte will also increase because of the high Km of the GLUT2 transporter. The increase in glucose will lead to binding of glucose to glycogen phosphorylase, which will shift the form of the enzyme from a relaxed state to a tense state. This change in, or this structural change in the enzyme will lead to a dissociation of phosphorylase and the regulatory subunit of PP1. Now phosphorylase cannot inhibit PP1 anymore, and now the PP1 will be active and be able to dephosphorylate phosphorylase, so it becomes less active, and it will also dephosphorylate glycogen synthase, where glycogen synthase will become activated. And that makes good sense, because when the concentration of glucose increases in the circulation, we're not interested in degrading glycogen, but we are actually interested in synthesizing glycogen. So, as I said before, glucose will lead to an inhibition of phosphorylase and activation of glycogen synthase simply by regulating the activity of PP1. And for those reasons, we actually also say that the phosphorylase acts as kind of a glucose sensor in hepatocytes. All right, so in order to understand how metabolism is regulated under, for instance, fasting conditions, we of course also have to understand how glycolysis is regulated in the liver, because we have to make sure that the liver actually produces glucose to the circulation and not degrading the glucose, because the liver, as I said initially, plays an important role in maintaining constant glucose levels under, for instance, fasting conditions. So in the liver, we know, as I said before, that glucose can enter the hepatocyte via a glucose transporter known as GLUT2, which has this high Km, meaning that the glucose is more or less in equilibrium across the plasma membrane. Then we know that glucose will be phosphorylated by glucokinase to glucose 6-phosphate, and glucose 6-phosphate can also be dephosphorylated by glucose 6-phosphatase. Once glucose 6-phosphate has been converted to fructose 6-phosphate, we know that fructose 6-phosphate can be phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase 1. On the other hand, fructose 1,6-bisphosphate can also be dephosphorylated by fructose 1,6-bisphosphatase. In liver, it's important to understand how glycolysis is regulated, and in liver, glycolysis is primarily regulated by the activity of phosphofructokinase 1, which is strongly regulated by a metabolite known as fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is a strong activator of PFK1 and an inhibitor of fructose 1,6-bisphosphate. So in order to control the level of fructose 2,6-bisphosphate, we need to understand how it can be produced or degraded. Fructose 2,6-bisphosphate can be produced from fructose 6-phosphate by being phosphorylated by another enzyme called PFK2. PFK2 is kind of a special enzyme because it can actually have two different activities depending on its phosphorylation state. PFK2 can be phosphorylated by protein kinase A, and once it becomes phosphorylated by pKa, it will actually change its activity from being a kinase to a phosphatase. So PFK2 is also known as the tandem enzyme, reflecting its two different activities. So in order to control the level of fructose 2,6-bisphosphate, we can actually control it by controlling the state of PFK2. And we know that under these conditions, under these fasting conditions, glucagon, as we saw before, will activate a G protein coupled receptor, or the glucagon receptor, that will stimulate the stimulatory G protein, which again will activate adenylate cyclase, leading to production of cyclic AMP and an activation of protein kinase A. Once protein kinase A is active, it will phosphorylate PFK2 and thereby changing its activity from a kinase to a phosphatase. This will result in less or lower levels of fructose 2,6-phosphate, meaning that we are actually removing a stimulator of glycolysis and removing an inhibitor of gluconeogenesis. So we overall will see that glucose is not being degraded, but actually we will be able to synthesize glucose via gluconeogenesis. We know that PFK1 can also be regulated by other means, for instance, the energy charge and citrate as in muscle, but these are less important in liver, where, as I said before, the far most important regulatory mechanisms by PFK1 comes from regulating the level of fructose 2,6-bisphosphate. Then we know that fructose 1,6-bisphosphate can further be metabolized in glycolysis to phosphoenol pyruvate, which of course also can be converted to pyruvate. And we have another control point of glycolysis here, where we know that protein kinase, a protein kinase can phosphorylate pyruvate kinase to its phosphorylated form and less active form. And this is also, or pyruvate kinase is also phosphorylated pKa, as we also already activated before. So simply by phosphorylating pyruvate kinase, we can also control how phosphoenol pyruvate is converted to pyruvate or used in gluconeogenesis. As we saw in muscle, pyruvate kinase can also be regulated by other means, by the energy charge or acetyl-CoA fatty acids or amino acids. As in muscle, we also know that fatty acids can be imported into hepatocytes by different mechanisms mediated by different transport proteins. And as we saw in muscle, the fatty acids will also need to be activated to acyl-CoAs in order to be metabolized. The acyl-CoAs will enter the mitochondria by the CPT system or the carnitine shuttle system, and also in liver cells enter beta oxidation, where the fatty acids will be converted to acetyl-CoA under the production of FADH2 and NADH. As in muscle, we also know that pyruvate can enter the mitochondria and be converted to acetyl-CoA by the enzyme called pyruvate dehydrogenase. As in muscle, we also know that the enzyme can be allosterically inhibited by NADH and acetyl-CoA, and also that NADH and acetyl-CoA will promote the phosphorylation of PDH to its phosphorylated form, which basically is inactive. So that will ensure that pyruvate is not converted to acetyl-CoA. And that is, of course, important, because pyruvate now can be converted to oxaloacetate. Also because acetyl-CoA from beta oxidation will activate pyruvate carboxylase, which of course will carboxylate pyruvate to oxaloacetate. We can of course produce citrate from acetyl-CoA and oxaloacetate, but we will also see that when, or in case that we fast more than 24 hours, the liver will start upregulating gluconeogenesis, which will lead to less, or which actually will use the oxaloacetate that we can produce from various sources and convert oxaloacetate to malate, which can be transported out of the mitochondria and become, which will be converted back to oxaloacetate. And then by PepsiK, oxaloacetate can be converted to phosphoenol pyruvate, which then can enter gluconeogenesis and lead to the production of glucose. So in case we starve more than 24 hours, we will see that the glycogen stores will be empty, more or less, and the liver now has to produce glucose by gluconeogenesis because it cannot degrade glycogen anymore, simply because the glycogen stores are gone. That will, of course, take away oxaloacetate from production of citrate. So when we starve more than 24 hours, oxaloacetate will be converted to malate and further enter gluconeogenesis. We still see that acetyl-CoA levels will be quite high because we keep degrading fatty acids in the liver. So in order to get rid of these acetyl-CoAs, we will see that acetyl-CoAs are converted to ketone bodies. And ketone bodies can now be used by, be secreted by the hepatocytes and into the circulation, where the ketone bodies now can be used as an alternative energy substrate in muscle, in the heart, and in the brain. So finally, we need to look at how fatty acid degradation and synthesis kind of can regulate each other. Of obvious reasons, fatty acid degradation via beta oxidation is quite important in order to understand how the energy expenditure takes place in different cell types. When we look at how fatty acid metabolism is regulated, we know that glucagon in liver can of course activate protein kinase A, and we know that fatty acids from the circulation bound to albumin can enter the hepatocyte and become activated to acyl-CoA esters. The acyl-CoA esters will enter the mitochondria, as we have seen a couple of times now, for instance, in myocytes and in hepatocytes, and they will enter the mitochondria via the carnitine shuttle system. In the mitochondria, the acyl-CoA esters, or the activated fatty acids, will be degraded to acetyl-CoA by beta oxidation, which also will produce FADH2 and NADH. When we look at how fatty acid synthesis takes place, then we know that in order to produce fatty acids, we need two substrates, acetyl-CoA and malonyl-CoA. And in order to produce malonyl-CoA, we need to carboxylate acetyl-CoA by an enzyme called acetyl-CoA carboxylase. So once we have the two different substrates, acetyl-CoA and malonyl-CoA, we can produce fatty acids. This, of course, as I said, requires that the enzyme, actually the rate-limiting enzyme of fatty acid synthesis, is active, and it's active when it's not phosphorylated. So acetyl-CoA carboxylase can be inactivated by phosphorylation. And this phosphorylation or phosphorylation of acetyl-CoA carboxylase can take place by the activity of protein kinase A. So once protein kinase A phosphorylate acetyl-CoA carboxylase, the enzyme will become inactivated. Then we also know that malonyl-CoA is an extremely strong inhibitor of the rate-limiting step of beta oxidation, namely the first enzyme in the carnitine shuttle system known as CPT1, or the carnitine palmitoyl transferase protein 1. So simply by phosphorylating acetyl-CoA carboxylase to an inactive form of the enzyme, we will produce less malonyl-CoA. That will, of course, result in less inhibition of transport of fatty acids into the mitochondria, thereby allowing oxidation of fatty acids. So if we are in a situation where we are fasting, glucagon levels will increase. That will eventually lead to activation of protein kinase A that will phosphorylate acetyl-CoA carboxylase, which will lead to less production of malonyl-CoA, which will then, where we then will remove an inhibitor of CPT1 that overall will allow our fatty acids to be degraded. This interaction between fatty acid synthesis and degradation is actually extremely important because of obvious reasons. It ensures that when we degrade fatty acids by beta oxidation in mitochondria, we're not synthesizing fatty acids. We also know that the activated form of fatty acids, acyl-CoA esters, are actually also an extremely strong inhibitor of acetyl-CoA carboxylase by itself. So simply by increasing the level of acyl-CoA esters will lead to an inhibition of acetyl-CoA carboxylase, leading to less production of malonyl-CoA, which basically will allow the transport of acyl-CoAs into the mitochondria via the CPT1 system. So I hope by this lecture that I have given you a short overview of how metabolism is regulated under, for instance, starving conditions. And I hope that you also can appreciate that there are some extremely important regulatory mechanisms, not only of the specific pathways, but also how these regulatory mechanisms actually can interact with each other, even between tissues. If you have further questions, I'm always open for questions, so you're more than welcome to contact me either by phone or email. Thank you very much.